underlying various infection-associated chronic illnesses, starting first with neuroinflammation, and then we'll explore the interplay between host, disease, and the microbiome through an omics approach. So then we'll move into examining what is known around the pathogen persistence hypothesis. So a lot is going to be said in this topic, and it's my great pleasure to introduce our first speaker, Andrew Miller. Andrew Miller is the William Timmy Professor and Vice Chair for Research in the Department of Psychiatry and Behavioral Science at the Emory University School of Medicine. He is an internationally recognized expert in interactions between brain and the immune system. I've been reading his articles for a long time, particularly in the role and the function of inflammatory cytokines during immune activation. So welcome, Dr. Miller.
Thank you, Brian. That was very kind. So we're going to switch gears a little bit and talk about the brain. I'm going to talk about the common mechanisms of behavioral changes that may be involved in many of the various disorders that we see, and the behavioral consequences that we see as a function of chronic infection-associated illnesses. And this work really got started with some observations that were made by oncologists, surprisingly enough, when they started using interferon alpha, the antiviral and inflammatory cytokine interferon alpha, for the treatment of malignant melanoma and other cancers. And later it was used, of course, for infectious diseases like hepatitis C. And what they noticed in these patients was there was a striking increase in behavioral changes in these patients. Whoops, went the wrong way. In these patients, unfortunately, those of you online can't see where I'm pointing, but you can probably guess. A substantial percentage of patients were showing a depressed mood, anhedonia, problems with anxiety, memory concentration problems, and then a preponderance of neurovegetative symptoms, including the most common symptom, which was fatigue, but also changes in sleep, psychomotor retardation. And for those of you who are in any way familiar with the criteria that we use to make a diagnosis of depression in psychiatry, we went in to see how many of these patients are becoming depressed. And this study here followed the patients for 12 weeks on high-dose interferon alpha, and after 12 weeks, almost 50% of the patients met symptom criteria for major depression. So this antiviral inflammatory cytokine was able to make these changes, these causative changes, if you will, in terms of behavior in this patient population. Now, just as a reminder, interferon alpha plays an important role in a number of infectious diseases, playing obviously a role in antiviral defenses, increasing OAS enzymes, RNase L, and degradation of viruses, activating innate and adaptive immune responses, and even playing some homeostatic functions in terms of the brain, having to do with synaptic plasticity and cognitive function. But what's interesting relative to our topic today is the fact that interferon alpha has also been implicated in a number of infection-associated diseases, including ME/CFS, where it's associated with increased RNase L and OAS2. There's also interferon alpha is associated with behavioral changes in HIV infection, being particularly associated with cognitive deficits as well as other behavioral symptoms. It's associated with increased symptoms of fatigue and cognitive deficits in Lyme's disease. And then in animal studies where animals are exposed to virus-type stimuli, interferon alpha in conjunction with other cytokines, notably IL-6, play important roles in regulating innate immune responses, neuroinflammatory responses, and then these sickness behavior responses, which were mentioned by Dr. Komarov earlier, which are very much like these same symptoms that we saw when patients started getting treatment with interferon alpha. What I thought was very intriguing is we were doing some studies with human microglia. These are induced pluripotent stem cell technology and deriving human microglia and then treating them with various variants of SARS-CoV-2 and found that there were differences amongst the variants in terms of the immune responses that were generated. And these are RNA sequencing data and gene set enrichment. And you can see a marked interferon alpha response in the Delta variant compared to Omicron and the Washington variants. And I think many of you are aware that the long COVID is more common in these with Delta variant versus the Omicron variant. And that's refer to that paper there. So there's this intriguing association with anti-inflammatory cytokines, interferon alpha, as well as inflammatory cytokines, and these behavioral changes that we see in patient populations. So how does this happen? What happens in the brain? And we've spent a ton of time. Those were early studies, as you saw before, on interferon alpha's effects on behavior, and then we've spent the last 20 or so years looking at the mechanisms by which these various cytokines are affecting the brain. And what we've learned is that one of the major targets of cytokines in the brain are the basal ganglia. These are subcortical structures that play an important role in motivation and motor activity. And so these are the ventrals, these are striatal regions, these are the basal ganglia regions right in here. And the ventral striatum plays an important role in motivation, in particular, the willingness to expend effort to go for a reward. And that decrease in that willingness is associated with a symptom called anhedonia, which is a core symptom of depression. And then there are dorsal regions of the striatum that are involved, and they're connected with more motor regions of the cortex, and these are implicated in fatigue, psychomotor slowing, decreased processing speed, cognitive dysfunction. And so cytokine effects on these areas are leading to these types of symptoms. And then it appears that what's underlying some of this is the cytokine effects on neurotransmitter metabolism. So disruptions in neurotransmitter metabolism ultimately disrupting neurocircuitry in the brain, the activity of these brain regions, then leading to these symptoms. And what we know is that among the neurotransmitters, inflammatory cytokines, antiviral cytokines can decrease dopamine, decrease its synthesis, its release, increase its uptake. So you see a decrease in dopamine, and it affects glutamate as well, but although I'm not going to talk about that today. So I'd like to just give you a little feel for this data. This is a motivational circuit here, and then the ventral part of the striatum, and the ventral striatum is this region right in here, tag of the caudate, lower tag here, the putamen and the nucleus accumbens, so that's the ventral striatum right in there. And to activate this brain region, we used a functional magnetic resonance imaging task and this gambling task where the patients got into the scanner, they saw 2 cards face down. We asked them to pick a card, or they picked a red card. They won $1, a black card. They lost $1. We rigged the game. Everybody won. They were happy. They got paid to be in the scanner, to be in the study. And surprise to surprise, if you look at the win versus lose contrast, in the sample as a whole, you see very nice activation in this ventral striatal region. However, if you then compare individuals who've been chronically treated with interferon alpha, this antiviral inflammatory cytokine, for 4 weeks in this case, you see that there's significant reduction in ventral striatal activation in the interferon alpha treated patients. These patients were getting interferon alpha for hepatitis C, and these were hepatitis C patients awaiting interferon alpha treatment. And you see this across the board, left, right, and bilaterally. As you might expect, these changes that we see in the win versus lose contrast and the activation of this ventral striatal area is associated, maps onto reduced motivation. So the higher, the lower the level of activation, the higher the level of reduced motivation in these individuals as measured by the multidimensional fatigue inventory. And you can see this in the sample as a whole, but you see a clustering of red dots here. And these are individuals with interferon alpha treatment who have both low activation in the ventral striatum and high reduced motivation or anhedonia. Now, what is important here is this isn't just something that we reported with interferon alpha. This has been shown with multiple other inflammatory stimuli. So this has also been demonstrated with endotoxin as well as typhoid vaccination given to healthy volunteers. So if you administer these inflammatory stimuli to healthy individuals, they too show reduced ventral striatal activation in association with behavioral changes, including reduced motivation and fatigue. So 3 different inflammatory stimuli having the exact same effects on the brain. This is a very reproducible, reliable finding that is seen across laboratories and across groups. Interestingly enough, we then tried this in collaboration with the CDC, and Beth Unger sitting here, who was, I think, the PI on this study. I think you were the PI. And so we did neuroimaging in these patients, and this is that dorsal part of the striatum that I was talking about before. And this is the caudate nucleus and the globus pallidus. We did the same gambling task in these patients with ME/CFS, and you see the same changes that we saw, or very similar changes that we saw with the interferon alpha treatment, significant reduction in activation in the win versus lose condition in the caudate nucleus in chronic fatigue patients compared to non-fatigue controls. You also see this in the globus pallidus, and this is associated, much like we saw before, with symptoms of fatigue. So here, a very strong relationship between the level of activation in the win versus lose contrast and symptoms, and this is in the globus pallidus, of mental fatigue, general fatigue, and reduced activity. And not much, we don't see much in the non-fatigue control population. So here we're seeing changes in the ME/CFS population that's very much like the types of changes that we see with individuals exposed to interferon alpha as well as other inflammatory stimuli, suggesting that this may be an inflammation-related phenomena. So what might be causing this? What might be a mechanism involved? Well, dopamine plays an extremely important role in motivation and motor activity. So we've looked at dopamine. In this case, we had to develop an animal model. We used a rhesus monkey model of cytokine-induced behavioral changes or inflammation-induced behavioral changes, and did in vivo microdialysis. That's a probe that goes into the brain, and we can sample extracellular fluid. 4 weeks on interferon alpha, similar doses to what is given to humans, and you see that in this same striatal region where we saw the activation changes in the brain scanning, we saw significant decreases in dopamine, extracellular dopamine, and its metabolite homovanillic acid. When we went in to then stimulate dopamine with either potassium or amphetamine, which is put in through the probe directly into the brain, you can see that there's significant decreases in dopamine release as a function of interferon alpha, and this decreased dopamine release was associated with decreased effort-based motivation. This, again, is a very reliable finding. It's been shown in laboratory animal studies with rodents, and it's also been shown, we've demonstrated this in humans using radiolabeled levodopa and PET scanning, and again showing this decreased release that maps onto decreased effort-based motivation as well as fatigue in these various species. So what about endogenous inflammation? What I've told you up to this point is giving various cytokine stimuli to people. What about inflammation that comes from within the body, which is most relevant to what we've been talking about today, because almost every speaker has talked about inflammation being a consequence of viral infection, reactivation, and the like. And to study this, we've used resting state functional connectivity. The patients get into the scanner, they stare it across, and we're looking at the relative activity in the brain in different brain regions, and brain regions that are firing in concert in synchronous fashion, we believe are talking to one another, and these then represent circuits in the brain, and we can then map those circuits onto behavior. And two of the major circuits we're interested in are the ones that you might guess are derived from the ventral striatum and the dorsal striatum. So ventral striatum to the ventral medial prefrontal cortex is the heart of your reward circuitry, your motivational circuitry, and then motor activity more related to fatigue, psychomotor slowing, these types of symptoms, as I showed you before, going up into these areas of the cortex that are more involved in motor activity. And we wanted to see what happened to the connectivity, what happened to these circuits in the brain that are involved in motivation and motor activity as a function of inflammation. And we did this in patients with major depression. In order to index inflammation in these individuals, we use CRP, which is produced by the liver. It's a very reliably measured inflammatory marker, and thanks to the American Heart Association and the CDC, we have indexes of what's low, medium, and high inflammation based on CRP. And so what we were interested in is, as your inflammation goes up, what happens to these various circuits involving both the ventral striatum to ventral medial prefrontal cortex and the dorsal striatum to more motor regions of the cortex. And what we found, actually we did a whole brain analysis, so when you do these kinds of analyses, you start at one place in the brain, you ask what's happening to the connectivity of this part of the brain to every other part of the brain. And the brain region that showed the greatest change in terms of its connectivity to the ventral striatum was the ventral medial prefrontal cortex. And the change we saw was a decrease in connectivity. So as your inflammation goes up, as measured by CRP here, your connectivity is going down between these two regions. In other words, your motivational circuits are crashing as a function of increased inflammation endogenously in the body. We also saw the same thing with these more dorsal regions of the striatum that are involved in motor activity and motor processing, fatigue, cognitive function, and connectivity up into these areas, and they also crashed as a function of increasing levels of inflammation. I should also mention that these changes that we saw in this connectivity mapped on to reduced motivation as well as increased fatigue and cognitive slowing in these individuals. So the circuits mapped onto the behavior. Now people wonder, and it's always something we get asked on grant applications and whatnot, well, how does peripheral inflammation, how does endogenous inflammation get into the brain? Well, actually, they've done studies on this, and there's an interesting recent study showing that for reasons that are not entirely clear to anybody, but in the context of stress and inflammation, there appear to be breaches in the blood-brain barrier that occur almost selectively in this same area of the ventral striatum and in the area of the striatum. And so seeing that data, we got very interested, well, what are the direct effects of inflammatory mediators on dopaminergic neurons? And so we've started studies, again, using induced pluripotent stem cell technology to derive dopaminergic neurons. And we treated those neurons with IL-6 and then did RNA sequencing and gene path analysis. And so we're looking here now at downregulated genes, and what you see is that the gene pathways that are downregulated are the ones that are involved in dopamine packaging and release. So chemical synaptic transmission, vesicle-mediated transport in the synapse, and signal release exocytosis. And interestingly, you can then treat these cells in vitro with a Janus kinase. Somebody mentioned something about JAK inhibitors. In this case, we use baricitinib, which blocks the signaling pathways that IL-6 uses to induce an inflammatory response. And if we take these IL-6 treated cells and add in baricitinib, and now we're looking at the upregulated genes, and you can see we can completely reverse these changes that we saw on packaging and release that I showed you before, the transport and the dopamine secretion and so forth. I should mention that baricitinib is one of the few drugs that is FDA approved for the use of acute COVID, and you can imagine what Lilly is doing right now. They've got a trial going on for long COVID using baricitinib, so it's very interesting that we have this. So let me just finish up with the summary here. Antiviral, anti-inflammatory cytokines. Our inflammatory cytokines lead to chronic changes in neurocircuits in the brain and neurotransmitter systems that contribute to neurovegetative symptoms, including anhedonia, fatigue, and cognitive dysfunction. And I think research priorities here should be to look at common behaviors that are altered in these infection-associated chronic illnesses. What is the circuitry that's involved? And what are the mechanisms of these immune changes on neurons and microglia and other other cells in the brain? And I think we're in a position where we can start to test pharmacologic agents. I like the JAK inhibitors in this regard for targeting these pathways that we know have effects on the brain and lead to changes in behavior. So let me stop there and thank you for your attention.
Our next speaker is Dr. Julia O, who is an associate professor at the Jackson Laboratory and a microbiome expert. Her work focuses on combining high-resolution computational reconstructions of the microbiome with experimental innovations to understand how host microbiome interactions result in disease, and then to use this information to develop microbiome-based therapeutics for disease prevention and treatment. So welcome, Dr. O.
All right, thank you so much for the invitation to present some of our works from the Jackson Laboratory here in Connecticut. Oops. So most of you know microbes in the context of infectious disease, because the conventional way that we think about microbes is that they cause different infections. So for example, COVID-19 virus, Helicobacter pylori. If you drink this bacteria like Barry Marshall did, it'll cause ulcers, or soil fungus causing lung cysts. And while these single pathogen to disease models of causality have been transformative in understanding infectious disease, over the last decade, our understanding of the role of microbes in our health reaches far beyond just infections. And this has been due to the recent research showing the role of the microbiome in our health. So the microbiome is comprised of trillions of different microorganisms, so bacteria, fungi, viruses, everywhere on our body. And rather than being infectious, they're overwhelmingly beneficial for our body, in fact, essential for our health. So they help us with digesting the food that we eat. They make vitamins that we can't make ourselves. They determine whether we can metabolize drugs as common as Tylenol, and they're absolutely critical in educating and maintaining our immune systems. And what we and others have found is that it's this repertoire of different microbes that we're exposed to cumulatively over our lifetimes that shapes how resilient we are to many different diseases, ranging from infections to autoimmune disorders, obesity, and even cancer. And just to highlight, there's been a really tremendous range of different diseases now linked not just to potential pathogenic microbes in the microbiome, but to broader changes in the microbiome as a whole. And central to how this notion of how the microbiome contributes to our health is this notion of homeostasis, where there's healthy microbes that beneficially regulate our immune system, and then those that can promote disease, whether it's by introduction of a pathogen like a virus, which has been implicated in chronic fatigue syndrome, or changes in the microbiome towards a pro-inflammatory configuration, which we can term dysbiosis. And the framework of our MACFS Center at the Jackson Laboratory has been to leverage high-resolution omics data, which is what I'll discuss today, to begin to predict some of these interactions to motivate mechanistic research, and at the same time to identify biomarkers that will allow us to better classify disease as well as potential agents of risk. And our central goal ultimately is to look at interactions of the microbiome with the immune system, so most likely mediated through metabolites produced or shaped by the gut microbiome, so for example, short-chain fatty acids, bile acids, amino acid metabolites, and others, and then examine how these interactions may be altered or dysfunctional in ME/CFS. Now, there's ample rigor of prior research that the microbiome can contribute to disease severity in ME/CFS. So, for example, infection can trigger ME/CFS, for example, Lyme disease, HIV, Q fevers. Fecal microbiota transplantation has had anecdotal success in ME/CFS treatment. Microbial dysbiosis had been observed in preliminary ME/CFS studies characterized by a loss of microbial diversity, a potential reduction in putative anti-inflammatory species, and potential increase in pro-inflammatory species. And similarly, there's precedent for metabolomic irregularities in ME/CFS. So here we're talking about circulating blood metabolites as being a reflection of what's produced by the microbiome that ends up in the bloodstream, as well as the activity of circulating immune cells, and then correspondingly the interactions with the microbiome and microbial metabolites. So for example, metabolomic irregularities have been observed in ME/CFS cohorts, including in lipid and fatty acid metabolism. These include decrease in sphingolipids and phospholipid and branched chain amino acids, both of which influence immune functions, including the inflammatory response. And then paired with that, in the blood, which was shown by our ME/CFS center head, Daria Unitmaz, immune dysregulation has been observed in ME/CFS, including changes to the frequencies, but also importantly, the functional capacity of different immune cell types, such as natural killer cells, and a variety of different T cell subtypes. And so we think there's a lot of collective precedent for integrating these omics approaches to generate new hypotheses for ME/CFS. And so the general framework that we have here is to generate very high resolution data sets for the gut microbiome, examining species composition, overall community structures, the functional coding capacity of the microbiome for different metabolites, and then pair this with untargeted plasma metabolomics and immune profiling. And then finally, what we do is integrate the most important data type, which is the manifestation of disease in these patients, which is collected with very detailed demographic data, clinical measurements, and survey questionnaires, all of which was performed by our wonderful collaborators at the Bateman Horne Center, Suzanne Vernon and Lucinda Bateman. So today I'm going to focus primarily on our efforts minus the immune profiling. We've generated all this data and are currently analyzing it and integrating it into some of our machine learning models. And so I'll just start out with a description of our cohorts. So we have this very unique design where we have these early versus late stage disease design. So about 75 patients in each stage, plus matched healthy controls. And we followed them longitudinally over time. We have that very detailed clinical and survey data that I mentioned, which we collapse into disease severity scores. We collect stool from metagenomic sequencing, which allows us to identify the microbial species, the genes they possess, and what metabolites they might encode. And then blood samples, which we perform plasma metabolomics on. And then we integrate all this data collectively with the clinical measurements, that microbial species and genetic data, and then the metabolomic feature analysis as well. And then, so just to summarize, we use state-of-the-art machine learning classifiers to then identify features which may come from any one of these different data types that are characteristics of the disease stage. So just the major take-homes of this work, which we published earlier this year in Cell Host Microbe. And I figured I would give these major take-homes up front so they could keep them in your mind as I go through some of the data, is that surprisingly, the short-term patients show the most significant microbial dysbiosis. And while the long-term patients had the most severe disease, they reestablished a kind of a healthy control, more look-alike microbiome, but they had the most major metabolic dysbioses in addition to the clinical disease. We leveraged this very high resolution omics data and the clinical data for a very robust classification of disease cohorts, both ME/CFS versus controls, but also the early versus late stage patients, really highlighting the value of this multi-omic analysis with these multiple cohorts. So just to give you an example of some of the data that we generated. So first, we examined microbial community diversity. So think the richness of the ecosystem, where loss of microbial diversity has been associated with numerous disease states. So here we're looking at richness on the y-axis with controls versus patients on the left. And then on the right here, the breakdown of the early versus the late cohort, where you can see that the early state has a much more pronounced change in the overall community diversity. We can be a little bit more specific and try to pinpoint specific microbes that are changing and contributing to the loss of this diversity. So looking at a very broad phylum level, at Firmicutes versus Bacteroidetes species, which are the dominant gut phyla. And then each of these lines is the differences of a species' relative abundance between the different cohorts. And so you can see that there's species that have significant differences in both, where there's a depletion of different Firmicutes in the patients, concurrent with an increase in some of the Bacteroidetes, which then normalizes in the long-term patients. Now, I'll go into a little bit more detail on these species in a minute, but we also identified that this ratio of Firmicutes to Bacteroides, which, like diversity, has been linked to different inflammatory diseases, was overall altered in the patient group, especially in the early stage. So just summarizing the statistic here. In contrast to the microbiome data, where we observed the greatest differences in the early stage patients, we saw the greatest changes in the late stage patients vis-a-vis the plasma metabolome. So again here, looking at the differences in the normalized abundance of different metabolites from three representative classes of metabolites. So a familiar face with the sphingomyelins, which you saw significantly increased in the late stage cohort, as well as diacylglycerol and phosphatidylcholine metabolites, similarly increased in the late stage cohort. And so summarizing then these initial findings, what we saw were microbial changes really in those short-term patients. But while those longer-term patients reestablish a microbiome that looks more similar to controls, they have significant differences in the blood metabolome. Now, we don't know why this is. So it could be that the short-term patients are iterating over many lifestyle and dietary changes, which we know shift the gut microbiome. Gastrointestinal disturbances tend to be more common also in the short-term cohort. So we don't know causality. But one hypothesis is that the accumulation of these early stage microbial changes could lead to longer term metabolic dysbiosis. And that would be a really interesting hypothesis to follow up on. So the next thing that we did was to see if we could use this data to then accurately differentiate patients from controls and also to identify features that were really specific to a cohort, because this disease is notoriously difficult to accurately diagnose. So we built state-of-the-art machine learning classifiers to model the ME/CFS versus a control microbiome. And this is a visualization of the test of the accuracy of these models, because this was called an ROC curve, where an area under the curve of 1 would be basically perfect. And that's a number shown in the parentheses there for all the omics combined versus each one of these different data types separately. And what you can see is that integrating all the data types brings us farther from that diagonal. So i.e. the most accurate in green in terms of differentiating patients is this integrated approach. We can then identify features or species or metabolites or clinical features that are really important to contributing to the accuracy of this model. So these are the top most important features. They're ranked by their importance to the model. And these were microbes that were known to produce different short-chain fatty acids or immunomodulatory amino acid metabolites. And so here colored by the predicted metabolic functions, and most notably these butyrates, which are in blue. Now, butyrates are of a lot of interest in the host microbiome immune interaction field. And these are micro-produced short-chain fatty acids, and they've been very strongly linked with numerous facets of immune regulation and function of a healthy gut, as well as a healthy gut epithelium. And what we're seeing is that the microbes that produce the butyrate, as well as the plasma butyrate, were significantly depleted, especially in that short-term cohort, as well as the long-term cohort. And so here, what we're doing is we're breaking down what species are contributing to this depletion. So showing full change across the cohorts in terms of the relative abundance of the butyrate-producing microbes, ranked here by the relative abundance compared to control, to show that butyrate-producing microbes are significantly depleted, so i.e. those smaller and greener circles. And so that brings us, so this brings us to next steps here, as well as knowledge gaps. And so I just wanted to summarize some of our data and then talk a little bit about potential research opportunities and kind of next steps to these studies. So while these really high resolution omics tools are really effective in terms of identifying potential disease markers, the next step is to take them to mechanism. So major limitations, however, in the field of ME/CFS, as well as I would venture to say other chronic diseases such as long COVID, is that no good animals model really exists because the disease is so heterogeneous and it has so many different physical manifestations. And so what I think that we need is that in human clinical studies, these need to be we need in human clinical studies that are paired with high resolution omics to identify causal links. And we can take some cues here from recent immunotherapy studies, which are relatively small interventional studies, but have been really effective in terms of leveraging the omics to basically create networks that have causal links. And so, for example, what might be feasible in an in-human clinical study, so for example, supplementation of some of these butyrate producers, so fecal transplantation or probiotic supplementation. Butyrate and other short-chain fatty acids could be administered as as a supplement, and then also dietary modulation to enhance the presence of these butyrate producers, which are often associated with consumption of fermented food. So these are potential studies that could be performed in ME/CFS patient cohorts, and then monitored over time to associate changes in clinical phenotype together with change in these omic profiles for potential causal links. And then finally, stay tuned for the integration of the high resolution immune data, which I think will similarly provide some of these causal links as well in terms of pairing microbial changes together with those changes in the blood metabolome that we observed. And with that, I'll thank my fantastic group at the Jackson Laboratory, primarily Royan Zhang, a very talented graduate student who performed all this analysis, and Ryan Caldwell, Elizabeth Fleming, who processed all the patient samples, and then the Unit Maz Lab, our ME/CFS Center, as well as our collaborators at the Bateman Horn Center as well.
Thank you, Dr. O. Okay, our next speaker is Dr. Bill Robinson. Bill is the James Raitt Professor of Medicine, Immunology, and Rheumatology at Stanford University, where his lab investigates the molecular mechanisms of autoimmune diseases, including work to define key roles for microbial triggers in the initial and progression of autoimmune diseases, such as the link between Epstein-Barr virus infection and multiple sclerosis. So welcome, Dr. Robinson.
Thank you so much, Brian. I appreciate the opportunity to participate and present at the symposium. And today I'm going to talk to you about the role of EBV molecular mimicry in multiple sclerosis. Great. EBV is an infection that affects approximately 95% of us and has been epidemiologically associated with MS, rheumatoid arthritis, Sjogren's, as well as systemic lupus. EBV infects B cells through the complement receptor CD21 and resides in the nucleus as an episome. And in most of us, approximately 1 in 100,000 B cells are latently infected with EBV, residing in the episome, where it expresses certain proteins that, like, tonically provide low-grade activation of the B cell. And on occasion, there's lytic transformation, where EBV reactivates and expresses the approximately 84 different genes in its genome and releases new viral particles. And lytic reactivation of EBV is associated with the development of MS, lupus, as well as clinical flares of lupus, as well as the development of long COVID, PASC. And so we're very interested in the role of EBV, not only as a molecular mimic, but also in the lytic reactivation driving autoimmunity. So most humans are infected with EBV when they're children or teenagers. And in many cases, particularly in the young, it's an asymptomatic infection. More in teenagers, it can result in infectious mononucleosis. And there's also certain cancers that are EBV associated. And as I mentioned, there's now epidemiologic evidence linking EBV with these different autoimmune diseases. But the overarching question is whether EBV is in fact a causal contributor to these autoimmune diseases or whether it's just happenstance and epiphenomena. So there was a paper last year from Alberto Ascherio's group at Harvard, and basically they showed that development of MS was inseparable from EBV infection. They had 801 patients that developed MS, and 800 of those patients had EBV infection prior to progressing to MS. So essentially 100%. And he'll tell you more about that work later today. There's reports from Judith James and John Harley that lupus is associated with EBV infection, that rheumatoid arthritis is also, as well as Sjogren's syndrome. So my lab wanted to further define whether EBV was in fact a causal mediator of MS versus just happenstance epiphenomena. And so we sequenced the B cell repertoires of spinal fluid B cells isolated from human MS patients. And you can see an example of this B cell repertoire from a human MS patient's spinal fluid here. And what's most notable about this repertoire is that it's highly oligoclonal, meaning that there's these very limited number of very large clonal families of B cells. So each of these red strips represents a family of B cells all sharing the same B cell receptor. And in the spinal fluid, you can see there's on the order of 10 to 15 different clonal families, which is very different than what we see when we sequence the peripheral blood. And we see an extremely diverse B cell repertoire where there might be only a few couplets or very small families. So to further define what these B cells were producing, we expressed the antibodies represented by the B cells indicated by the arrows. So basically by a representative B cell from all the different major clonal families identified. And we spent several years trying to figure out what these antibodies bind. And because on our first passes, they did not bind robustly to our human MS myelin arrays. And eventually, Toby Lance put these on our viral antigen arrays. And we identified this antibody MS39 highlighted here that potently bound to EBNA1. And further, when we made EBV tiling arrays, we found that this antibody MS39 bound to what we term a hotspot region on EBV in EBNA1, 385 to 400. And so we became very interested in this antibody because this region was previously described as related to molecular mimics in both MS as well as in lupus. So just zooming in on this region of EBNA1 where this MS39 antibody, it's this proline arginine rich region with a core at 394 to 399. And when we express the germline version of the antibody, so the mutated version has this higher affinity and the germline a slightly lower affinity, suggesting that affinity maturation of the B cell was essential to have robust binding to this EBNA1. We did staining of brain sections and found that this MS39 antibody stained brain sections in a glial pattern, as you can see here. We put them on human protein arrays, and the top hit was GliaCam. And GliaCam is a glial cellular adhesion molecule that regulates water and chloride homeostasis. And it's expressed by both astrocytes and oligodendrocytes, and thus immune targeting of GliaCam would disrupt the myelin sheath and thus provide a mechanistic basis for how it could mediate clinical symptoms of multiple sclerosis. So when we did fine mapping of GliaCam, we found that there was this core epitope region that was shared between EBNA1 as well as GliaCam, and you can see it here with prolines and arginines. And further, we found that in contrast to EBNA1, where the antibody we isolated from MS spinal fluid bound with a very high affinity, it bound to native GliaCam with a significantly lower affinity. And we came to appreciate that GliaCam is phosphorylated in the myelin sheath. And when we tested against this phospho-GliaCam, the form present in the human myelin sheath, we see a much higher affinity of binding of our MS39. indicating that in human MS, this antibody that we isolated was actually against the post-translationally modified phosphorylated form of GliaChem. So thus far, what I've shown you is that EBV infection activates anti-EBNA1 B cells, and it must do so repeatedly to cause affinity maturation, resulting in somatic hypermutations in the B cell receptor that result in gaining reactivity to this phosphoGliaCam that can then target and bind the human myelin sheath in the brain. So is this reactivity pathogenic or not? And so we ran the mouse model of multiple sclerosis, the EAE model, the MOG-induced model. And when we co-immunize with this EBNA1 peptide versus a scrambled peptide, what you can see is that the mice immunized to develop EAE exhibit high titers of anti-EBNA1 reactivity, and that they gain reactivity to GliaCam over time. This is the cross-reactive GliaCam epitope. And further, these mice that gained, that were immunized with EBNA1 and gained these GliaCam cross-reactivities exhibited a more severe course of EAE. They were more paralyzed as compared to mice that received the scrambled peptide, suggesting that these anti-EBNA1 GliaCam cross-reactive responses can contribute to demyelinating disease in mice. So this is looking at larger cohorts of human MS patients and comparator patients, and you can see the very high titer anti-EBNA1 antibodies in MS patients, including a quarter or so of patients that exhibit reactivity to this key region of EBNA1, 386 to 405. So this is antibody reactivity in sera. And we further show that these same patients exhibit high reactivity in their serum antibody response to myelin GliaCam, and that we can cross-block. So basically, if we do cross-blocking, we're looking at antibodies to EBNA1 in these same patients, and when we block with EBNA1, it blocks the binding, and when we block with GliaCam peptides, it also blocks the binding, suggesting it's the same antibodies that cross-bind to both EBNA1 and to myelin GliaCam. We also see evidence for a robust cytotoxic CD8 T cell response against EBNA1 and myelin GliaCam. So this is like a quantiferon type of assay, but done with EBNA1 or GliaCam. And you can see in human MS peripheral blood, a high level of granzyme B CD8 T cells. So these are cytotoxic T cells, both against EBNA1 as well against GliaCam, both the extracellular and intracellular domains. We also saw CD4 T cell responses against EBNA1 and GliaCam, as indicated here, both Th1 as well as Th17 type responses, with the Th17 responses being more prominent. So thus, what I've shown you is that EBV infection can induce activation of anti-EBNA1 B cells, and that repeated activation and trips through the germinal center, likely through EBV reactivation, result in the affinity maturation of the encoded antibodies to cross-bind to myelin GliaCam, and that these cells activate a reciprocal T cell response, particularly a cytotoxic CD8 T cell response that also targets phosphorylated myelin GliaCam, which could mediate CNS and myelin tissue destruction and thus result in the symptoms of MS. So where's all this action happening? This is the amino acid sequence of EBV EBNA1. This dark gray shaded area is the DNA binding domain. EBNA1 is a transcription factor, so this binds the DNA. And all the mimics are located in this junction between the disordered and structured region that's outside of the DNA binding region. And so zooming in on this region, what you can see is that we call this the mimicry region or a hotspot region. Here's our GliaCam mimic at 394 to 399. And it happens to overlap with Judith James and John Harley's Smith mimic for lupus. So this just adjacent overlapping region is the mimic for anti-Smith autoreactivity in lupus. There's another MSEA antigen just upstream here. And Thomas Olson's Anoctamin 2 mimic for MS is just further upstream here, all within a close proximity of this disordered structured junction within EBNA1. And we're very interested in the biochemical properties by which this could mediate molecular mimicry. So just to summarize, our data, we believe, demonstrates a critical role for EBV EBNA1 mediated molecular mimicry, driving the autoimmune response that underlies potentially the pathogenesis in approximately 1/4 of human MS patients. And we think that it's likely that there's additional mechanisms of B cell and other immune cell activation that contribute both molecular mimicry as well as potential other mechanisms that are responsible for the mechanisms by which EBV could contribute to the pathogenesis of, additional subsets of MS, as well as lupus, RA, Sjogren's, and long COVID, and potentially other syndromes that have a chronic autoimmune and fatigue component. So with that, just recognizing the people in my lab, Toby Lance and Camille Brewer really drove this work. Toby Lance is a newly minted assistant professor at Stanford, and we work in close collaboration with Larry Steinman on all of our work. With that, happy to take any questions at the appropriate time.
Thanks, Bill.